And good afternoon. How's everybody doing today? Nice to see you. Cool. Looks like we got a lecture going on here. I don't know if we've got anybody logged in today. I'm going to wait for just a minute, see what we got going on here. Let's see if we've got a good lecture going. If you're in this lecture, go ahead and type your name in the chat bar and let me know how you're doing. Go ahead and type in. Let's see. We got it right here. Okay. Fantastic. Oh, there we go. Nalisa's here. Hi, how are you doing? Good to see you. Who else do we have here in the lecture today? It's a good thing you showed up today, Nalisa. Hope you're doing well. Fantastic, fantastic. I'm glad you logged in. I was getting ready to switch to a recorded video. If you hadn't showed up, I was going to close down this broadcast. Do you have any questions so far, Nalisa? How are you doing today? You can make it today. Fantastic. Okay. Well, I'll go ahead and do oh, hello, Cosette Reeflin. Holy cow, Melissa. This is Cosette. She is one of my former students. Was it this summer or was it this past spring, Cosette? She's stopping by to say hello. What you'll find is a lot of my students will, from time to time, bump into a lecture just to say hello. And so that's Cosette coming in to say hello today. She was one of my great students. Wow, it was last fall. Has it really been that long, Cosette? God, it's almost like I remember you from yesterday. But she was nice enough to come. Cosette, I'm here with Melissa, and I'm getting ready to get started with my lecture on biopsych. I changed the class completely around, Cosette, so you wouldn't even recognize it. But I'm going to go ahead and get started if we could. Okay, so today it's Module 3, Ghost in the Machine. Oh, thank you. That's awfully awesome, Cosette. What we're going to be talking about today, actually for the next two lectures, we're going to be talking about this idea of the human body and the soul. So the question is, when we finally come down to try and talking about the human being, we have to understand that there are some things that happen that are very, very hard to document and to demonstrate physically. And so, actually, one of the famous philosophers, there's this problem in clinical psychology, actually in philosophy. The debate was called the dualistic debate. The idea was, do human beings have a soul? Right? Or are we simply driven by the atoms and the chemicals and the molecules that we have in our body? Now, if you were one of these people that thought that human beings had a soul, you might be called a dualist. You argue that there's a soul that lives inside the body and controls the body. And other people, more mechanistic, monistic people, or materialistic, would say, you know what? There is no such thing as a soul because we can't see it, we can't measure it, so it must not exist. And so, what you're going to find in psychology is there's this big debate that's existed for thousands of years about whether or not there is such a thing as a soul, and how is it that this soul, which is non corporeal, which doesn't really exist, can move something that exists in 3D space. And so, one of the things that psychologists argue about, and Melissa, this week in webinar, I'm actually going to show you a couple of videos where current neurophysiologists and neurophilosophers talk about this question. Is there something, some sort of dynamic that exists over and above the system that controls who you are? And when this question comes to consciousness, we often have difficulty because consciousness, one of the things that psychologists still haven't figured out, not only what it is, but how to even measure it. And what I'm going to do is this idea of figuring out the soul is what we might call the hard problem in psychology. Okay, and it's one of the things that psychology can't answer yet. It's the question of the future. And the hard problem is, what does it mean to feel like being a human? What is a soul? Where is consciousness? And why does it feel so magic? The easy problem we're going to talk about is this mechanistic problem. I can show you how information comes in your eyeball and goes all the way to the back of your brain. And I can show you exactly how that process occurs. Now, it wasn't easy per se, but scientists were able to figure it out. And so I can explain to you exactly how vision works. That's the easy problem. The hard problem, on the other hand, is to explain what does it mean to look at a sunset? What does it feel like when we look at a sunset? Why do some people like this painting and not that painting? That's the hard problem. What does it mean to be a human being? And so this week we're going to ask the question, what exactly does it mean to be a human being? I'm going to start today by talking about the physical part of the debate. We're going to talk about the things we know, the easy problem, if you will. And then on Thursday, we're going to focus on the hard problem a little bit more. Okay, so the easy problem starts in the branch of psychology we'll call biopsychology or neuroscience. All right, and so this is one area of specialization. If you remember back from week 1 when I talked about all the different modern subfields of psychology, biopsychology is one subfield where you can become a biopsychologist and study the brain and nervous system. And biopsychology is where the easy problem starts. And that's chapter 3 in your textbook. Okay. Now, the nervous system. The nervous system is the part of your body, the human body, that perceives your environment and controls your body. You can think of nervous system as the organs and structures in the human being and the animal, although we're just focused in humans, that allow the human or the animal to receive, interpret, and respond to information in the environment. For this lecture, I'm only going to talk about the brain and the spinal column. Okay, on Thursday, we're going to talk about the parts of the nervous system that are called the peripheral nervous system, which is the nerves that run to the distal parts of your body and send messages back to your brain and spinal cord. But your brain and spinal cord are generally considered to be the control centers. They're in charge of how you feel, how your body works, how you think, how you behave. Those are the control centers. Your brain controls your muscles and your internal muscles and your internal organs. It controls your voluntary behaviors. And your spinal column is like an old brain that is responsible for reflex behaviors. If any of you have ever been to the doctor and you've been in the doctor and they hit your knee with that little rubber hammer and your leg moves, that's a reflex. Reflexes are actually controlled by your spinal column. So we've got these two control centers of our body, the brain and the spinal column. We'll call those the central nervous system. Now, what you're going to find in the nervous system is that there are two broad types of cells that make up the nervous system. The first are neurons, and the second are support structures called glial cells. Now, your neurons are the nerve cells that contain everything you are and make you do everything you do. These neurons work electrochemically. All right. So each of these neurons is sensitive to chemical stimulation. So you can pour little chemicals around the neuron and it'll cause that neuron to generate electricity. And neurons create brief pulses of electricity called action potentials. Okay. And so neurons communicate by means of electricity and chemicals. Okay, now generally speaking, we are born with all of the neurons that we will ever need, and they're put into the right places in your nervous system, and they're already mostly connected. All right, there may be 100 million of these neurons in your nervous system, and their job is to transfer information from one spot, my head, all the way to my fingertip. That's what neurons do. They communicate information, and they do so with chemicals and by producing electricity. Now, surrounding all of these neurons, these 100 million neurons in your central nervous system, are surrounded by these things called glial cells. Glial is a Greek word for glue. These are the glue cells. And the job of the glial cells is to support the function of your neurons. They help your neurons work faster. They speed up the communication of your neurons. They clean up intracellular waste. They're like the garbage men in your nervous system. And they also insulate these neurons and keep them from touching one another. Neurons generate electricity. And just like wires, electrical wires have to have insulation on them, so do your neurons, okay? So in your nervous system, we've got 100 billion neurons. And these neurons are sensitive to chemicals, and these chemicals make them generate electrical potentials. And the support structures are called these glial cells. And these glial cells insulate them so that your brain doesn't short circuit. And they speed up the electrical impulses that travel along your nervous system. Now, you'll notice in this picture underneath me, they've got two different kinds of cells. They've got oligodendrocytes and astrocytes. There are actually half a dozen different types of glial cells, but in an intro psych class, they typically don't talk about what the individual glial cells do. So I just want you to know that they're kind of the supporting structure. So when we start looking at a nervous system, we have to understand that it's composed of these two parts: neurons that communicate and glial cells that support and insulate this electrical generating organ that you have in your brain. Okay, let me make sure I didn't go da 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 da. Now, here's the deal. For those of you who are interested, what I would like for you to do is see if you look down at the very bottom of this link, there's a page. You'll see there's a link to Neuroscience for Kids. Now, I found this link several years ago, and you know what? There's nothing kiddie about this. The reason they call it Neuroscience for Kids is because it's not filled with all kinds of jargon that only neuroscientists understand. However, if you have a question about the nervous system, what glial cells look like, what action potentials sound like, what happens when a black widow spider bites you, if you want to learn all of this stuff about neurons and the nervous system, I highly suggest you go look at Neuroscience for Kids. It's going to help you answer some of the questions even on the homework or the quiz. Okay? Now, the human nervous system, as I mentioned, it operates neurochemically. We talk about the neurochemistry of the nervous system. Neurons don't touch. These electric producing neurons don't touch. Instead, they're separated by really small microscopic spaces called synapses. And the reason this is the case is so that when one neuron generates electricity, it doesn't continue going. It stops when it gets to the end of that neuron. Now, what it causes to do is it causes that one neuron to dump the chemicals that are into that neuron into this space called the synapse. And these chemicals bathe the next neuron and cause it to generate electricity. So if you look at this picture underneath me right here, you see a neuron. This is a neuron. Now, this neuron, you'll notice it has these tree-like structures called dendrites on the one side. These dendrites come up to and almost touch another neuron. And what happens is these dendrites have these receptor sites that fit neurochemicals perfectly. So when this neurochemical is secreted from the first neuron, it floats into this synapse and it goes right across this synaptic barrier and it interacts with the next neuron. Okay? And what it does is it causes that neuron right here. Okay, this neuron is being bathed in chemicals. If it gets enough chemical stimulation, this neuron's going to generate a pulse of electricity. This pulse of electricity is called an action potential. And what it does is it runs from the cell body all the way down the length of this thing called an axon to the terminal buttons, the other end of the neural cell that holds chemicals that it's going to release. Now, here's the deal. An axon can be microscopically long, or there are actually axons. There are nerve cells in your brain that are a foot, that have axons that are a foot and 2 feet long, that go all the way from your brain all the way to the bottom of your spinal cord. Okay. And what this means is chemicals can be secreted in one part of your brain right here, and they interact with the dendrites. The dendrites cause a pulse of electricity, which travels to another part of your body where the terminal buttons are filled with chemicals. These chemicals, once they're stimulated by electricity, these chemicals are released into another synapse, which can cause another neuron to begin producing electrical pulses. And this is how your nervous system communicates. So you have a thought up here, and it causes this neuron right here to generate a pulse of electricity. And that pulse of electricity comes all the way down to somewhere else in your body to the terminal buttons, and it causes another neurochemical to be released. And then that neurochemistry causes another neuron in a different part of your body to generate activity. So my brain can send a message down to the axon in my arm, and then the axon in my arm, when it receives that chemical message, it will then raise itself. And so, in a sense, you've got these neurons, and they have these little chemical sensing tree-like fibers called dendrites. And when those dendrites are activated by a chemical, they cause an electrical impulse to go all the way down the length of that axon to release chemical in another part of your body. Now, one of the things I want you to notice in this picture right underneath me is that we have this stuff called myelin sheath. Now, when you get a bunch of glial cells, they will wrap themselves around the length of an axon, just like insulation on electrical wire. And when you get all of these glial cells together, they're known collectively as the myelin sheath. So this myelin sheath is composed of lots and lots of glial cells, okay? Now, here's, if you look over on this chart right here, what you're going to notice are some of the main neurotransmitters that are used in your body. It turns out that there are about 50 chemicals produced in your nervous system that can affect the way neurons act, okay? 50 different neurotransmitters. However, the 7 that I have right here in front of you do probably 95% of the work in your brain. So when you are thinking and really focused on something, your brain is generating acetylcholine in this frontal lobe to help you focus and think. And when you learn something and you are able to remember something for a test, glutamate is being released in your brain. So glutamate and acetylcholine are really responsible for being a good student because they cause your brain to pay attention and they cause your brain to be able to remember things. Okay? Now, if you've ever had somebody tell you something that makes you feel really good about yourself or something good happens, you are going to experience a release of endorphins in your brain. Endorphins are sometimes called your body's natural painkiller. Endorphins were the first neurotransmitter discovered way back in the 1950s, and they were called endorphins because it was seen as an endogenous pain-killing substance. They said, wow, this is like an endogenous kind of morphine. Morphine's a painkiller. Put those two together, endogenous morphine, you get endorphins. And your body releases endorphins whenever you feel good. Dopamine. Dopamine has been implicated in Parkinson's disorders as well as in schizophrenia. Now, if you don't have enough dopamine in your system, dopamine kind of makes your motor movements nice and smooth. My father has Parkinson's disease, so he shakes a lot and his hands move when he's trying to sit still. So he has to take this medicine called levodopa, and he eats this medicine levodopa, and then when it gets to his brain, it's metabolized into dopamine. So people with Parkinson's disease have to take extra dopamine, and this dopamine smooths out their behavior and makes them able to have smooth motor control. Okay, now, uh, now there's another neurotransmitter. Have any of you ever been familiar or been around and heard anybody talk about being an anxious person or having an anxiety disorder? People who have anxiety disorders take drugs that magnify the GABA in their system. GABA is what we might call it's an arousal decreasing kind of neurotransmitter, and so it causes you to relax. People who have anxiety have to take Xanax, and Xanax magnifies the amount of GABA in their system and causes them to relax. Okay. Now, if you look down here at the bottom, serotonin. Serotonin has often been implicated in people's moods. We often think that people who are depressed don't have enough serotonin in this system. That's known as the serotonin hypothesis. However, I will tell you, research has come out in the last year saying that depression is way more complicated than just the absence of serotonin in your brain. However, serotonin is somehow implicated in people's ability to maintain a positive mood. So, what I want you to understand is that you've got these 7 chemicals, and they are contained in the terminal buttons of your neurons. Now, not each neuron will have different kinds of neurotransmitters in it. So each neuron will have one type of neurotransmitter in it. So you might have dopamine in this part of your brain, you might have serotonin over here, you might have acetylcholine right here. So there are different accumulations of neurotransmitters in your brain. And so when stimulated, different parts of your brain will release these neurotransmitters, okay? And so we have different kinds of neurotransmitters. They're waiting in different parts of your brain for that nerve to be stimulated. When that nerve is stimulated, it releases that chemical, which causes other neurons to do other things, to become active and to do other things for you. Now, I do want to mention this one point before we move on, this idea of neuroplasticity. Here's the crazy things. As I mentioned earlier, your neurons, your neural architecture is pretty much in place even before you're born. Okay? However, when you stimulate a nervous system, you notice how these dendrites kind of look like roots and these terminal buttons look like roots? Well, here's the crazy thing. When one neuron stimulates another neuron, it's just like fertilizing a root. And what happens is when you stimulate a neural connection, these neural connections will begin growing together. And on the other hand, if you don't use a neural connection, just like if you don't water a plant, what happens to the roots? They wither and die away. So the crazy thing about your brain is it comes with this general architecture, all right? But as you grow up and use different parts of your brain, these brains are going to start to grow together and knit together. And there's actually this concept in psychology we call a critical period. In order for different abilities to develop normally, that part of your brain must be stimulated at a critical period. So, in order for your eyes, your vision to develop normally, you have to use your eyes during the first 6 months of your life. Now, of course, I use my eyes during the 6 months of my life. But what psychologists have done is they've done these studies with animals, and they find if you cover an animal's eyes up for the first few months of its life and then take the covers off after that animal has reached a certain age, the animal will be functionally blind because that part of the brain didn't get stimulation with chemicals, and the little dendrites and terminal buttons didn't grow together. I don't know if you know this, we're going to talk about this later, but we actually have two areas in our brain that help us speak. We have what's called a motor center, which helps my lips and my tongue move. And right behind it, I have a grammar center, which helps me put words together in a meaningful sense. And then in my temporal lobe, I have a dictionary of all the words I can say. In order to talk, my motor area has to move my mouth, right? My memory area has to have the right words to use, and my grammar area has to put all of those together in the right order. Here's the crazy thing about language. Anybody can learn language as long as they are exposed to it in the first couple of years of their life. There are some examples of children who were brought up without language for the first 3 or 4 years of their life, and these children never ever learn how to speak. And that's because this part of the brain didn't receive stimulation, and so the brain actually reorganized itself to do another activity altogether. Does that make sense, folks? To give you a good example of this, if you look in this week's course resources folder, there's the story of Ben Underwood, the blind kid who sees with his ears. With his ears. Here's the thing. He lost his eyes due to cancer when he was a really, really young kid. But his mama told him, you can still see, you just can't see with your eyes. And so this kid, he got into the habit of clicking and making sounds. And crazily enough, one day he began to be able to see kind of like a bat with this is the blind kid. No, that's not a metaphor. Look it up. This kid can actually, he uses acoustic sonar. The information comes in his ears, Nelisa, comes in his ears. And because his eyes didn't work, his eye center grew and it invaded his hearing area. And so now, whenever sounds come into his ears, they're processed by his vision center. And this guy, Nelissa, actually can visually imagine the world because he can hear echoes just like a bat. It's insanely crazy. This guy rides a bike, he plays basketball, he goes to school and plays football, he kicks his brother's butt in video games. You have to watch this video, Nelisa. It will blow your freaking mind. And it is a perfect example of neuroplasticity. He had a brain area that wanted eye information, but it didn't get any information because his eyes had been removed due to cancer. And so that part of his brain needed something to do. So it started growing, looking for something to do. And it was stimulated by the acoustic area, his part of his brain that hears sounds. And these two areas grew together through neuroplasticity. And now he can do what bats can do. Amazing, amazing, amazing. So definitely check it out. But what I want you to know about brains is you come with a prepackaged brain that has a certain set of modules, if you will, and has a certain set of activities. However, if you're missing a body part or a part of your brain is damaged, your brain can actually rewire and reprogram itself so that you can still use it. How amazing is that, right? Your brain comes so that if you are missing a part of your body, it can rewire and still be normal and let you have a normal life. I had a student in my class one year. This was probably about 10 years ago. I was giving this neuroplasticity lecture. She raised her hand and said she had grown up completely normal, but when she was a young adult, she started having headaches. And she wound up at a neurologist's office. They did a CAT scan and found out as an adult she was missing her frontal lobe, her whole left frontal lobe. She was missing part of her brain. And yet, for all intents and purposes, she was completely normal. She'd been a normal student. She drove a car. She walked. She didn't even know she was missing part of her brain because neuroplasticity had caused these neurons to rewire themselves. So think of your brain as having 100 million wires that connect your control center to your most distant parts of your body so that your brain can receive information. Ooh, somebody's touching my finger. And so your brain can send messages. Ooh, it's time to move the finger. And this all happens two ways. Our brain releases chemicals called neurotransmitters, and these neurotransmitters cause neurons to generate action potentials or pulses of electricity. Now, here's the crazy thing. I don't know if Nalissa, I see we have a couple people in here beside Nalissa. I don't know if any of you have ever drank alcohol before, or smoked marijuana, or taken any other drug. But the reason that drugs work, the reason they call them psychoactive drugs is because these drugs can get through the blood-brain barrier, they can get into the brain, and they are shaped the same way as neurotransmitters. And so they sneak into your brain and they start doing the job of neurotransmitters, but they don't do that correctly. Okay, now Lisa, if you've ever, let's say you drink coffee, actually it turns out caffeine is a neuromodulator as well. The caffeine molecule gets into your brain, it passes through the fatty blood barrier and gets into your brain, and it acts at the acetylcholine receptor sites. Say, oh, green tea has got even more caffeine. So when you drink caffeine and it kind of wakes you up, Melissa, that's because it's activating this frontal lobe. It's acting in the place of acetylcholine. And so we call psychoactive drugs, we call them neuromodulators. If you're depressed and you take depression medicine, if you're anxious and you take anxiety medicine, all of these are what we would call neuromodulators. They modulate or change the effect that these serotonins. That is exactly why you feel awake. Absolutely. So it's going in there and it's activating the neurons in your frontal lobe. That's why I drink tons of coffee. As a student, a lifelong student, I always had to stay awake and focus. So I learned how to drink tons and tons of coffee. Now, if you'll go to that website that I showed you, Neuroscience for Kids, there's a cool little blog on neurotoxins, poisons that work by messing up the chemical balance in your brain. So, if you've ever been bitten by a spider or some other animal, they have neurotoxin. There are two kinds of things: neurotoxins and hemotoxins. Hemotoxins break down the blood and the blood vessels in your system, but neurotoxins cause you hurt you by causing your brain to do crazy things. So when you get bit by a black widow spider, the latrotoxin, which is the poison of a black widow spider, it actually works at your acetylcholine receptor sites, causing you to feel pain and muscle cramps. And that's sort of where that neurotoxin works. And so any poison gas that you've ever heard about works. If you've ever watched CSI or NCIS, Melissa, and seen them talk about a poisoning victim, all of these poisons and toxins work by working in the nervous system, these neurotoxins. Okay? Now, it turns out that knowing all about the anatomy and physiology of the brain is relatively new. Your mother watches those shows on 48 Hours. She knows exactly what I'm talking about then. Yep, there's always some sort of secret poison that somebody's using. and investigators always got to figure it out. Now, just like you're talking about 48 hours in NCIS, psychologists are also investigators too. And the problem with neuroscience until the 20th century is we didn't have computers, we didn't have nuclear medicine to look inside living brains. And in fact, until the beginning of the 20th century, we didn't even have refrigerators. So when a person died, we couldn't even really open up that brain and spend a lot of time looking at that brain because it would rot and atrophy, right? So the invention of the cooled mortuary chamber, the invention of electricity and computers, the invention of nuclear medicine has allowed us to actually begin looking inside living brains. Now, before all of that was apparent, the only way we could really learn about brain damage about the brain was by looking at people who had brain damage. And so what I'm going to do over the course of this semester is I'm going to present you with lots of case studies about people with brain damage. I don't know if you remember my last lecture, I showed you Phineas Gage, the railroad worker with the iron spike that went up through his brain. That railroad spike destroyed his frontal lobe and his personality changed. So the case study of Phineas Gage led people to suspect that your personality and your planning and your impulse control is controlled right here in the frontal part of your brain. Okay, there's a very, very famous French doctor named Paul Broca who had a patient that couldn't speak. That his patient, this was in the 1800s, his patient couldn't speak. The only word his patient could say was the word tan. And so that was his nickname for him, Tan. And after Tan died, Paul Broca did a autopsy and found that Tan had damage in his frontal lobe right here on the side. And so this area, known as Broca's area, we began to realize that this was responsible for speech, right? Now, when Broca figured that out, he thought that this was where all speech was created. But then a fellow named Carl Wernicke had a patient that had damage a little bit farther back, about 2 inches, in a different part of the brain called the temporal lobe. And this patient could speak, but couldn't use or understand grammar. And so what he realized was that Paul Broca had discovered the motor part of language, the part that makes your lips move together. But then there's another part of your brain that puts the words together in a grammatical sense, right? And so we learned all of this from people who have brain damage. So one of the things that we might have learned from Ben Underwood, the blind boy who sees with his eyes, we've learned that actually the brain has this quality we'll call plasticity. It can rewire itself. So case studies have historically been the way we've learned about the different parts of the brain. However, with the advent of electricity and nuclear imaging, we've begun to see inside of active brains. And so we're going to talk about CT scans, MRIs, fMRIs, and PET scans. Okay. So what I'd like for you to do is learn a little bit more about PET scans, CT scans, and MRI scans. So if you're looking underneath me, what you see, these are two. If you look on the left, that's A is a CT scan. Actually, I think these are both CT scans. And what you'll notice is that we can look inside and we can sort of see the structures inside the brain. Now you'll notice on A, you'll see that big ring around the outside. That's the skull. And then inside you see these gray areas, and they are the different areas in the brain. Now brains are very, very symmetrical. And so if you look at B, you can notice that B is not very symmetrical because on the right side, there's this big shadow right underneath me, and it's called a tumor. So looking inside with a CT scan, we can actually see what's inside the brain. Now, the crazy thing is with this thing called a PET scan, a positron emission tomography, we can actually see what part of the brain is active at any given time. And so what they'll do is they'll put somebody in a PET machine, a PET scan machine, and then they'll give them, if it's symmetrical, then it's considered, I won't say it's a healthy brain, but lack of symmetricality suggests non-health. How about that? You can have a symmetrical brain and still maybe have problems. But when we're looking for tumors and anomalies in the brain, if we don't see if we don't see symmetricality, there's probably a problem. But don't think that symmetricality necessarily means healthiness. But that's a good way to think about it. That's a good way to think about it. Okay? So we have brain imaging. PET scans allow us to see what part of your brain is active at any given time. Okay? And it turns out that and we'll actually talk about that on Thursday a little bit more. And then when we get to sleep on Thursday, we're going to talk about EEG recordings. As I suggested to you earlier, Melissa, your brain generates electricity. And so what we can do is we can put these electrodes, I think it's 20 of them, all on the outside of your brain. And then when your brain generates electricity, we can measure the wave form of that electricity. And this has been used in sleep research to prove that when you are dreaming, your brain is more alive and active when you're dreaming than when you're awake. Isn't that crazy? And so when people go to sleep at night, you can use an EEG to show that people's brains slow way down and then speed way up, and then slow way down and speed way up. And we learn this from EEG recordings of the electrical activity in people's brains. And over the course of the day, depending upon what you're doing, your brain activity is going to change. When you're sitting here focused on my lecture, Melissa, your brain's going to generate one type of electricity. When you sit back and chill and watch TV and aren't paying attention, your brain generates another kind of electrical activity. If you ever sit down and meditate trying to reach a higher plane, Melissa, your brain will generate still another kind of electrical activity. And we can measure this all from the EEG recordings. And so the science of neurophysiology has come a long way in probably the last 50 years because of the invention of computers, nuclear medicine, and other cool imaging techniques. Ooh. That's a good question, Nelisa. It would depend upon whether or not you're just sitting back and the music's in the background, or are you focusing, really trying to understand the words and how it makes you feel. If you're really, really focused on the music, your brain is probably just like you were dreaming. But if you got just background music on and you're just kind of vegging out, probably it's going to be less active. That's a really good question, Nelisa. Very good question. Now, yeah. So using these techniques, case studies, cutting open and doing autopsies, PET scans, MRIs, EEGs, these other things that are beyond the scope of this class, like cell staining techniques, diffusion tensor imaging, all of these other ways have allowed us to really figure out what's going on in the brain. And so what I want you to appreciate, Melissa, is that your brain isn't one computer doing one task. Your brain is actually like 100 really small computers. And each one of these computers is doing its own program, right? And your consciousness or your final awareness is the combination of all of these little computers working, doing their own thing. So this part of my brain is moving my lips. This part of my brain is doing grammar. This part of my brain is remembering the words that I was supposed to say. And this part of my brain right back here is allowing me to look at this screen and see what I'm supposed to be seeing on this screen. All of these different parts of my brain are just like little machines doing their own activity, and they don't care what else is going on. And so the reason we know this is because when you destroy different parts of people's brains, an entire activity won't go away. It'll just be changed a little bit, right? So if you have these two areas, you can speak and do quite well. If you take away the frontal area, Broca's area, I can't speak, but I can still understand what you're saying. If, on the other hand, you leave Broca's area intact but destroy Wernicke's area, I'll move my mouth, but everything that comes out will be complete gobbledygook and won't make a bit of sense. And that's because each of these computers is doing its own thing. And so you can think of the brain as being a series of modules, independent modules. This part of my brain helps me focus. This part of my brain remembers things. This part of my brain moves my left foot. This part of my brain helps my eyes see. This part of my brain way down here breathes for me. This part of my brain up here tells me when it's time to go to sleep. So I have all of these modules working together. Does that make sense, Nalisa? Now, the neat thing is, if you look at the brain, what you're going to notice is it has some features that help us to understand its organizations. Now, we have a left side and a right side of our brain. As I suggested to you, Nalisa, a brain it has this very, it's this duplicate look to it, right? So you've got this even on left side and on right side. Both sides are absolutely the same. They're symmetrical. You have a left side of your brain and you have a right side of your brain. Here's the crazy thing. We have this thing called the contralateral design. Nobody knows exactly why it happens, although we think that sometime during the first couple of weeks of gestation, all brains seem to go into this twisting motion. But here's the crazy thing. The right part of your body, right part of your brain controls the left part of your body. So my left arm is controlled by the right side of my brain. My right arm is controlled by the left side of my brain. My left leg is controlled by the right side of my brain, and my right leg is controlled by the left side of my brain. Check this out. What I see on the left side of my visual field is actually processed in the right side of my brain. And what I see on the right side of my visual field is actually processed on the left side of my brain. And so for every brain area you've got, there is a duplicate, a left side and a right side. So symmetricality is a really good way to tell if a brain is normal, Nalisa, although it might not always be that final, that indicative. All right. Now, so you can divide the brain into a left and a right. Okay. And then if you look in the middle picture, we can organize it into three areas based upon what these jobs do. So the hindbrain part of your brain, it was the first part of the brain to evolve. And it's the same part of the brain that we share with lizards and snakes and frogs. I call it your lizard brain, if you will. This is your hind brain. And this is the part of the brain that's going to help you move. It's going to help you breathe. It's going to keep your heart beating. All of the things that lizards can do that you do are controlled by your hind brain. Okay. Now, then if you look on the top, we have this thing called a forebrain. And this is the part of the brain that evolved latest and is most pronounced in humans and primates. This is the forebrain. And your forebrain controls your conscious awareness, okay, right? And then you have a part of your brain called the midbrain, which connects the forebrain and the hindbrain so you can think and make voluntary decisions to do this or that, to pay attention to this or that. Okay? And so as we move from back to front, you go from more animal-like control behaviors to more human-like control behaviors. And the part of your brain that causes you to pay attention and focus and be a good student or a great lawyer is this part of your brain called the prefrontal cortex, which actually developed very, very last in the evolutionary line. And this controls the most human of all your behaviors and abilities. Okay? Now, look underneath me on this third picture. You can then go down to the cortex, okay, which is the forebrain part that I showed you earlier. And we're going to be on one side, the left side and the right side. What you can do is you can go into the left side or the right side, and using these fissures, these sort of creases in the brain, you can separate the brain out into four lobes. What we call a frontal lobe, what we call a temporal lobe, what we call a parietal lobe, and what we call an occipital lobe at the back of your brain. And each one of these lobes is functionally independent. You can take a live brain and well, a dead brain, of course, if you got it, and you could actually pull it apart. And so it's going to be like four parts put together on each half. So you have two frontal lobes, two temporal lobes, two parietal lobes, two occipital lobes. You have sort of 8 lobes of your brain, 4 on each side. This is going to control your focus and your motor movement. Deciding to get up and walk across the other room will occur here. Your temporal lobe is responsible for memory and hearing. Your parietal lobe tells you where you are in space. Melissa, right now I know you are not sitting next to your car, but point to where your car is relative to where you are sitting in space. I can tell you that my car is below me and behind me because I know where I am in my space. I can do that because my parietal lobe is working well. And then your occipital lobe, its major responsibility is for vision. Okay, it's to the far left. The reason you know that, Melissa, is because your parietal lobe is working. Here's a crazy, and I can show you this on Thursday night when you come to webinar, Melissa. Um, uh, there are people with damage to their parietal lobe, and what you can do is you can hold their keys, car keys, or an object up in front of them, and they can tell you what they're looking at. But then when you ask them to grab it, they can't find it in space. You put it right in front of them, and they won't be able to reach it because their brain doesn't know where it is in 3-dimensional space. And that's what your parietal lobe is responsible for. Now, on your own, Melissa, I want you to look at the function of each lobe to find out what they can do. So what happens in the frontal lobe? And there are several things. You got personality, you got smell and taste, and moving your fingers and hands in your frontal lobe. Your temporal lobe is for speech, for hearing, and for memory. Your parietal lobe is going to give you a sense of touch and a sense of space. And then your occipital lobe is going to be primarily focused on vision. Okay? Now, you know what? We were about to run out of time. We do have one thing I do want to mention to you pretty quickly is we talked at the beginning of the lecture about the easy problem and the hard problem. And so I'm going to talk to you a little bit about the easy problem and show you how we can actually map a sense and how we know how senses work. Okay, now sensory receptor cells, they're not in your central nervous system. They are actually on the outside part of your body. And we call them sensory receptor cells because their job is to turn sensation from the outside world into patterns of electrical activity inside your brain. So a light photon comes inside your eye, Melissa, and your photoreceptors, your visual receptors, turn that light energy into action potentials that are interpreted in the back of your brain. When you hear my voice coming in your ears, Melissa, the hair cells, which are your hearing receptors inside your ear, the hair cells bend and they turn the sound waves into patterns of electrical activity that are interpreted by your brain. Same thing when I touch you. The pressure that you feel on your arm is turned by your nociceptors or your mechanoreceptors into patterns of electrical activity that you're in your brain. So when you think about sensation and perception, basically what we have to do is we have to turn everything in the world into a pattern of electrical activity that our brain can interpret. And we have mechanoreceptors that are responsible for turning pressure energy into patterns of electrical activity. We have chemoreceptors that turn chemicals into patterns of electrical activity. We have photoreceptors that turn light energy into patterns of electrical activity. We have thermoreceptors that turn the heat of the environment into electrical activity. And we even have special receptors that tell us when our body has been violated by something dangerous. We call those nociceptors. And all of these are neurons that turn the outside world into information that your brain can interpret. When we get dizzy, I've heard what happens when our brains, when we get dizzy. All right, right. It turns out, I don't put these down here. This another receptor, another set of senses is your cerebellum, and that's located inside your inner ear. And you have these hair cells inside your inner ear here. You have these semicircular canals, Melissa, one in each plane of the world. So you got one that goes up this way, one that goes in X plane, one goes in the Y plane, one goes in the Z plane. And those hairs, those semicircular canals are filled with fluid and hairs. And as you turn your head to the left and the right, that fluid runs around in these three canals and sort of tells you which way your head happens to be facing. So when I turn my head this way, the fluid in my semicircular canals changes position and it rubs those hairs a different way. And then that information then travels down to my cerebellum at the back of my brain and tells me what position I am relative to gravity. That's a sense of balance, if you will, right? And so, Melissa, your sense of balance can be damaged by a lot of things. If you drink alcohol, that's a good one. I'll answer that in a minute, Grace. Here's the deal. Sometimes, Melissa, people get sick and they get a head congestion. And if you get all this fluid build up in your head and your ears, you know how your nose is connected to your ears through the eustachian tube. Sometimes people get dizzy because the fluid buildup in their ears prevents the semicircular canals from working properly. And that's why they get dizzy, right? And so you could think of that as what we would call a peripheral reason for being dizzy. On the other hand, Melissa, sometimes people will drink a lot of alcohol. And alcohol is a neuromodulator. Remember, it's a psychoactive chemical. And what it does is it puts your cerebellum to sleep. And so if you drink a lot of alcohol, you will also lose your balance. Now, that's a different reason. The information is here in your ears. The semicircular canals are working properly, but when they send that information to your cerebellum isn't working properly. So in order for us to have balance, the semicircular canal has to be working properly, and the part of the brain that interprets that sense has to be working properly as well. Does that make sense, Nalisa? Same thing with the visual information. I could damage your vision by taking your eye out, okay? Or I could damage your vision by taking your occipital lobe out. Either one of those is going to damage your vision. One's peripheral and one's central. Grace Trinidad, why do people like spicy food if it causes pain? You know, taste is a very bizarre kind of combination. And the receptors that give you pain for hot food are different from the ones that give you pain from like getting stabbed or something. In your tongue, you have these taste buds. You folks know the taste buds, and you have these taste receptors inside your buds. And you have, I think, like 50 or 60 different taste receptors. and they all combine, or actually, I take that back. You have 5 basic tastes, but they can combine to give you a complex combination of tastes. Why people like to have the sensation of burning might be the hard problem, Grace. At the beginning of class, I talked about the easy problem. I can tell you how taste gets from the tongue to the brain. Why some people like spicy versus salty. That's a tough question. Ah, good question. Good question. Okay, you know what? I am about out of time, so Nalisa, Grace, I'm going to let you finish up the last couple pages by yourself. And in, I'm sorry, that wasn't a great answer, Grace. I went to answer it and I realized that it's why people like spicy food is an awfully difficult question. On Thursday night, when we get in class, I'll talk a little bit about the visual system of vision, and I'll show you some really cool brain damage cases to kind of help you understand what we're talking about with regard to these different parts of the brain and this idea of brain plasticity and brain modularity. I am so sorry for running out of time. I would like to thank both of you for coming to class today. If you have any questions, send me a text or an email. Thank you again, Melissa and Grace, for being here. You are truly appreciated. I didn't get through as much as I wanted to say, and I apologize for that. So I'll try to do better next time. But if you don't have any questions, I'm going to go ahead and stop this broadcast. If you do want to talk to me later, don't hesitate to send me a text or an email. Oh, you're the best, Nelisa. You're just too nice. All right, folks, y'all have a great day, and I will see you on Thursday. Bye.